Thank you. Thank you for a very kind introduction. Well, first, let me do a sort of advertisement. So I'm from University of Maryland. Here is my department. So we do pharmaceutical sciences. You can see here, so we have 28 faculty members in this department. And we do very basic science from the very basic disease molecular targets to until clinical trials. We have a GMP facility and we have a variety of other centers. So we welcome collaboration from any industry partners and also foundation. So here is an example. For example, so we do here for the drug discovery for renal fibrosis from very beginning, lead identification, AI, directed, design, selection of candidates, and we do preclinical, we do, ADMET, and formulation. So we have faculty from very beginning basic science to the clinical part, as I mentioned. So MedLab, so MedLab is a translational research lab. So MedLab do very typical academic drug discovery development. So for drug discovery, so we are working on a particular pathway, one single pathway, and also AMPK pathway. So we studying drugs, we discover drugs based on transporter mechanism. And we are also working in the therapeutic areas in the metabolic disorders and cancer. So in terms of drug development, so we're working on membrane transporters, how those transporters can be utilized to better design your drug. And also we can do basically assays, so there are FDA guidelines for transporters. And we can test all those transporters for your specific substrates with our cellular assay systems. So we have expertise techniques here, so we can do drug property assays, we can do cellular molecular assays, and also we do preclinical mouse model. We can do PK studies, toxicity evaluation, assessment, and we also do clinical research. For example, on genotyping, biomarker analysis development, and so on. So today, so my talk is focused on liver fibrosis. Basically, it's actually derived from the metabolic associated fatty liver disease and steatohepatitis. Previously, so the MA, SLD, and MASH, so they got another name. So it's called non-alcoholic fatty liver disease and non-alcoholic steatohepatitis. So now people tend to use those new terms. But basically, so here, so the epidemiology, data. So this is a very prevalent disease in human populations, also including in the United States. And the fatty liver disease basically is relatively mild, so it can be reversible. So if, for example, so if you change your lifestyle, you change your diet, so the fatty liver absolutely can be reversed to normal. But so this problem is, many people, they're resistant to do the diet change and also the exercise, for example, also lifestyle change. So those fatty liver disease can be progressed to MASH or to the steatohepatitis. And in this case, so their fibrosis developed, and also further, so it can be further progress to cirrhosis and can affect a variety of other metabolic disorders. So, but this disease is basically, in overall, it's a silent liver disease. It's got, once you have the steatohepatitis or MASH, so your risk for more severe liver disorders and metabolic disorders can be dramatically increased. So what the status of right now for MASH treatment? So before 2024, there is no FDA approved drug at all. But luckily, so since 2004, there is one drug got approved. It's called the resmetirom. And it's a thyroid hormone receptor beta agonist for MASH treatment. And this year, another drug got approved. So it's semaglutide. So it's a GLP-1. We know it's a very popular drug right now. So it's for body weight loss, but also their clinical data suggest that the drug can be also used to treat MASH or fatty liver disease, steatohepatitis. But those drugs, so it looks like it's a done deal, right? So when the news come out, so my students actually ask me, so we're done. So, but actually, so if you look at the details, the data, the clinical data, for both drugs, well, even they got pretty good efficacy in terms of steatohepatitis. Basically, it can reduce the fat accumulation in the liver very much. But on the other hand, so if you look at the fibrosis, so the majority of patients, they couldn't get improvement in terms of fibrosis. So the better of this field is not done yet. So still, we need to look for, novel therapeutic modalities. So now, here, so what actually is the challenges of MASH drug development? So for a long time, people believe so the MASH, the pathogenesis is because of the two hits theory. So basically, you got the first hit is probably because of the obesity. So obesity as a early marker for lipid accumulation in the liver, and then so you probably got the second hit. For example, you got very stressful, got other HIV, or those not HIV, the liver virus infection and so on. But since 2016, so people believe the theory got a little bit changed. So people think for the MASH, for liver metabolic disorders, it's got to be involved in multiple pathways. So the multiple pathways work synergistically to aggregate the hepatic damage. So, and another challenge for MASH drug development is because in the clinical, it's really actually not easy to diagnose the fatty liver or the steatohepatitis or the fibrosis. So you need to do the biopsy, which is not good for the patient resistant to it. And the other way is, like I mentioned, is because of the multiple pathways involved in the disease. So the MASH is, like I said, it's a silent disease. So developing therapeutics adhering to the multiple hit hypothesis is actually really a priority right now in the field. So now, so let me walk you through the avenues. So we got this, series of a new compound. So my lab, so we work on the metabolic disorders quite a while. So beginning probably more than 15 years when the genomic GWAS studies got very hard in our field. So the genomic-wide association studies. So those GWAS studies, they identified many risk genes for a variety of diseases. For example, for the diabetics, they identified those, those different genes that are, risk genes for diabetics. And we worked on those, genes. So for example, those particular gene called the TCF7L2 or the protein called TCF4. So which is a transcription factor. And this gene is actually the most with the highest risk for the diabetic diabetics. So we lock out the mice we knock out the gene in the mice. So our data and the two other groups we found. So if you knock out the gene in the liver, for example, so you got the significantly reduced lipid accumulation in the liver. So this data showing here, so it's had dramatically reduced lipid accumulation in the liver tissues. So my lab, so at the beginning, so we were interested in actually the drug-induced metabolic disorders. So in the clinical, so you probably know, so there are many drugs. So if you take it for a while, so you can develop metabolic disorders. The very typical example is antipsychotics. Antipsychotics is if once you use it, you need to use it for quite a while, and the people really don't like it because once they take the drug for months, and then you got significant body weight gain, and along with other metabolic side effects. So we're thinking, for those drug-induced metabolic disorders, maybe there sort of there crosstalk between, the drug and those diabetic risk genes or metabolic risk genes. And those risk genes, you got polymorphisms, you got mutation, probably you're not gonna, have a increased risk for those metabolic disorders or drug-induced metabolic disorders, but also may change your response to those drugs that can be used to treat a metabolic disorder. So we work on this hypothesis. And we actually did a clinical study, for example, for olanzapine, which is antipsychotics. So we did find those diabetic risk polymorphisms, they can affect the response to the olanzapine. But the question is, so then we think, well, if you got, those diabetic risk genes, so they can, increase the risk for metabolic disorders, and those polymorphisms can affect the drug response. And if, for example, in our study for the T772, which is a Wnt signaling gene, and it turned out for the majority of those diabetic risk genes, so they're somehow either direct target or somehow related to Wnt signaling pathway. So that's why, so we began to work on Wnt signaling pathway. So then we went to look at literature and try to identify, if we can actually use the Wnt signaling pathway inhibitors to treat the metabolic disorders, because those increased Wnt signaling activity seems related to an increased risk of metabolic disorders. So we went to the literature and we find this particular drug called pyruvilium. So it's a potent Wnt signaling inhibitor and identified by Ethan Lee lab, and the mechanism they claimed is via the CK1 for activation and also axon stabilization. But the issues with pyruvilium is it's got very little or even no bioavailability. So the pyruvilium has been approved by FDA for the treatment of pinworm infections. So pinworm is in the GI. So you take the drug, so the drug doesn't have to cross the intestinal wall and can have a pharmacological effect against the pinworm. But for the systemic application, for example, to treat metabolic disorders, we gotta need a drug that can be able to cross the intestinal wall. Well, even by using the IV injection or IP injection, so we did see sort of metabolic benefits with pyruvilium, but we still think pyruvilium probably not good for metabolic disorders treatment. So the bioavailability is a big issue, and also pyruvilium seems quite toxic. So in the cellular assay, so we did find that the drug can cause significant cytotoxicity. And also the compound is not that stable. So it's got very light sensitive and also very unstable in acidic or even also basic environment. So the question is, can we develop novel analogs that can basically retain the Wnt signaling inhibition capability, but with a better drug properties? So here is our work. So we develop an assay. So Wnt signaling activity report assay. So we use that assay to screen compounds. So we collaborate with my colleague, Dr. Fengtie Xuan. He's a medicinal chemist. So he modified the compound pyruvilium, and we have more than 50 compounds, and we screen them against the report gene assay for Wnt signaling activity. So we'll be able to find, for example, several compounds, for example, YW1128, YW2065. So they got dramatic Wnt inhibition potency. And also we found, so just consistent with the reporter, the literature, so the compounds can stabilize the axon, which a key player in the Wnt signaling pathway. And we also found that the compound can reduce the protein level of beta-catenin, which is another key player in the Wnt signaling pathway. And on the other hand, so we found that our compound can, besides Wnt inhibition, it can actually activate AMPK pathway, which I'm going to talk about a little bit later. So here, so how we actually identify that our compound can also activate AMPK. So we found, well, first of all, so our compound can stabilize axon. So axon is a scaffold protein. So it's a key player, like I said, in the Wnt signaling pathway. So it can bind to a variety of proteins to form a protein complex. And this protein complex, can be degraded by a proteasome. So if you got axon stabilization, so basically, so you can it can bind to another transcription factor, beta-catenin, and lead to increased degradation. So in other words, so you're gonna reduce the level of beta-catenin, and beta-catenin is a transcription factor, so it can regulate a variety of genes transcription. So if you stabilize, if you increase the axon level, so you're gonna decrease the beta-catenin level and eventually lead to Wnt signaling inhibition. But like I said, when axon is a scaffold protein, so it can not only bind to axon, but bind to beta-catenin, but also can bind to other proteins. So in the there is a reporter. So when we look at the literature published in Cell Metabolism and also Cell paper, I think 10 years ago, and they found so the axon can also bind to AMPK. So it can, by binding to AMPK, so it can, lead to increased phosphorylation of AMPK by its upstream kinase called ALKB1. So then, so we tested, if our compound can, have a dual activities, can inhibit Wnt signaling pathway at the same time can increase the AMPK activity. So AMPK is a very important protein. So it's basically a cellular energy sensor in our body. So upon activation, so it can do a variety of things. For example, increase the fatty acid beta oxidation and reduce the lipogenesis. And for example, the overall, so it has metabolic benefits. So and since our compound can inhibit Wnt signaling pathway at the same time can activate AMPK, so we tested if our compound can have any metabolic benefits. So this is the first generation of the compound, so the YW1128, like it showed in previous slides. So it can inhibit Wnt signaling. So we tested the compound in vivo in the mouse model. And it seems the compound did have therapeutic efficacy against liver metabolic disorders in the high-fat diet-treated animal model. So it can reduce the body weight somehow. It can dramatically reduce the lipid accumulation in the liver, and also along with a couple of other metabolic benefits. So the Wnt signaling is also important in cancer. So probably some of you know it. So for example, in colon cancer, so more than 90% of patients, they actually got upregulated Wnt signaling pathway. So we think, maybe our compound can also be used to treat, some cancer. And we tested in a colon cancer animal model. It turned out our compound showed efficacy against the colon cancer growth in the animal model as well. So, but the parent compound, the 1128 and 2065, they're not good enough yet. The reason is, their solubility is not that great. Their bioavailability, even, it's much, much better as compared to the paravinyl. Paravinyl has got almost zero bioavailability. But for 1128 and 2065, it's got about 15%. But it's not good enough yet. So when we do the animal studies, we have to dissolve it in the in oil. So for the chronic treatment. So at the end of the treatment, so there's so much oil actually in the body, so which is not good. So Dr. Xuan further optimized the compound, and we got our very recent lead compound YA6060. So it's got very dramatic increased drug properties. For example, for the PK, so the bioavailability now is being improved to more than about 75%. And in the mouse study, so it's the half-life it's not that bad. So it's got about 1 hour and a half, and about 2 hours actually. So it's also seems very safe. So we already completed the acute toxicity assessment and look at those different tissues and looks like the drug can be well tolerated in the mice. So for the single treatment, the mice can tolerate up to 1,000 milligram per kilogram. For 5-day repeated doses, the mice can tolerate up to 500 milligram per kilogram. So it's pretty good safety margin at this point. So we're still doing the safety or the toxicity assessment in disease models. Hopefully, the drug eventually could be safe. So moving forward, so we did further assessment, efficacy assessment in animal models. So here is the very recent data. So it looks like the compound can somehow reduce the lipid accumulation, which is actually not as dramatic as we saw with the parent compound 1128. But what we really excited is, it looks like the compound can reduce the fibrosis very much. And this, encourage us to further test, our compound, if our compound can actually reduce the fibrosis in animal models. So we use, first you use the cell culture, the model, so LX2 cells. So it's a hepatic status cells. And you can see here, so the compound can dose dependently reduce those fibrosis markers. For example, the collagen 1, the vimentin, and the alpha SMA. And further, so we of course also validate if the compound work through the axin stabilization and also the AMPK activation. So the take-home message from this slide is, we lock down the axin 1, for example. We also activated the beta-catenin and locked down the AMPK. And we did see the results as we expected. It looks like our compound can have those actions through those Wnt signaling and AMPK pathway. So the most important part is, we want to see if there is any in vivo efficacy for our compound. So here, so we use a fibrosis model. So we treat the mice with the DMNA, which is chemical that can induce dramatic liver fibrosis. And we treat our compounds in this disease model. So what you can see here is the results is very dramatic. So it looks like the fibrosis got very significantly reduced as compared to the DMNA treatment alone. So, and also in the in vivo situation, we validate the pharmacological mechanism. So you can see here, so the drug treatment can basically stabilize the axin level. Meanwhile, can reduce the beta-catenin level, and also along with the winter inhibition expectation. So the two Wnt signaling target genes, cyclin D1 and c-Myc, their mRNA expression got significantly reduced with the drug treatment. So we also try to see, well, even we know so our compound can stabilize axin and can activate AMPK, but what's the direct target? So we're still wondering, so what is the direct target? And we synthesized those biotin-labeled compound, and we use the proteomics to try to identify, which protein could be the binding partner for our compound. And we'll be able to identify, a variety of proteins, of course, but the most significant one is protein T. Well, our, technology office don't want me to release the name of this protein. But anyway, so here, so we did some cellular mechanistic validation. And if we lock down this protein, so we did see the increased AMPK activation. And we also, if we lock down the protein, we did see increased axin. Meanwhile, so if we lock down the drug, so we did see the drug effect on the axin got abolished as well. So our compound, so not only actually, so in terms of fibrosis, not only have efficacy against liver fibrosis, so here is the most recent data. So we did see very exciting results that our compound can also reduce the renal fibrosis as well. And here is again, so in terms of those markers for fibrosis and also for the mechanism, so we did see our compounds as, effect. So here is a summary. So basically, so we found, so our, YA660, the parent compound have exhibited pharmacological activities in treatment of diet-induced metabolic disorders and colon cancer. And our compound has been discovered to possess excellent drug properties. And the active, the analogs can inhibit Wnt signaling and activate AMPK pathway. It has exhibited impressive efficacy against not only liver fibrosis, but also renal fibrosis as well. So here, my lab, so we, of course, the work has been done by students and postdocs, and we have collaborators and funding as well. Thank you for your time.